to get started with the first session of the morning, which is on controlling neural circuits. And so these talks will span sort of issues of control of brain networks or brain circuits in a number of different contexts. And I'm glad that we're going to open with Garrett Stanley, who I think has been a thought leader in this area for a number of years from Georgia Tech University.
Okay, well, thanks. I'm going to start my timer here. So thanks. I'd like to first thank the organizers here and for putting together a really wonderful meeting. I think I'm really excited just about the content. I think my career has kind of brought me to this point. I'm very excited. And also excited to see some old friends. And I'm going to be presenting some work. In the spirit of a workshop, I'm going to present work that's all unpublished. So we'll see how that goes. But what I'm going to talk about is closed-loop control in the thalamocortical circuit. And this is work done by a graduate student in my lab, Michael Bolus, with some help from Clarissa Whitmire, and also with Adam Willett, who is co-advised by Chris Rosell. It's a big collaborative group we have to work on these problems, and we have support from NIH and NSF. So the general kind of problems that our lab works on is in trying to have a two-way conversation with the brain. So we're trying to understand how information is represented from the outside world in sensory pathways, and part of that is reading the neural code, listening to the brain, monitoring activity from groups of neurons and trying to understand and make sense of this. But the flip side of this is writing of the neural code, of imprinting activity into the circuits in the brain so that we might communicate with the neurons. So in this spirit of this two-way conversation, we have a good conversation that's bidirectional. And we've done a lot of work in these two areas, but recently we started to get into pulling all this together. So it's really exciting to have this meeting kind of embody this for us. So what's our goal? A goal is fast, millisecond, robust control of neural activity to drive complex trajectories that ultimately lead to perception and guide behavior. So the motivation, probably don't need this, but the motivation for this kind of work is that patterns of activity in the brain are very complicated, and when we measure this in naturalistic settings, they're very complicated and creating a difficult demand for writing in signals. And that we think that, much like Steve gave a wonderful introduction of a lot of the issues that we face as well, we think that closed-loop control could also be used as a investigative tool, much like Hodgkin and Huxley used in their investigation of the nonlinear dynamics of action potential generation. We think that we can broaden that way of thinking to more complex circuits. And we also, it turns out that ongoing activity patterns in the brain are very influential of percepts and sensory pathways. Lots of examples in the literature of very context-dependent activity that gives rise to very different kinds of percepts depending on the ongoing activity in the brain. So there's some really challenging control problems here. And then I think we have a number of talks in this meeting that will get into some of the more therapeutic applications in DBS, for example, where commercially available devices are now, in addition to being able to stimulate, are actually made and able to record activity. So I think it just opens up a lot of problems for all of us to really tackle here. So what are the real challenges here? Well, fast millisecond control, heavy demand on hardware. So we've got filtering, A to D conversion, D to A conversion, et cetera, and software, spike sorting, model implementation, algorithmic control, and so on. And these are all really big challenges, and we've got to turn this around really quickly when we're closing the loop on these kinds of circuits. We have imperfect sensors and actuators, really imperfect. They are getting a lot better, and the field is exploding in this direction. So I think there's opportunities now that may not have existed 10 years ago just because of our ability to interact. Complex noisy neural dynamics that adapt and change. If there's a fundamental signature in the brain, that things change, and that's a real challenge in a control problem. And then changes, and maybe an extreme version of this is changes in brain state. Fundamental changes in dynamics from moment to moment, depending on what people in neuroscience often refer to as state. So what I'm going to talk about today, I'll try to keep this tight because I have this 30-minute chunk, is the model system that we work on, the framework that we're developing, some basic results in open and closed loop control, and I'll unpack some of the technical details of this for you, and then address what I think are some interesting issues in robustness, and then get into some very, very recent work that we've been doing on what we refer to as state-aware control, where we're actually respecting these transitions in state of the brain activity. Okay, so the model system we use is the rodent vibrissus system. It's strangely enough, it's turned out to be a very powerful model system in neuroscience, where I don't know if you can see, sensory inputs come in the periphery. Rats and other rodents have whiskers on the side of their face they use to palpate the outside world, and movement of the whiskers induce deformation of soft tissue in the face, and there are mechanoreceptors that send signals to the brainstem, to the thalamus, to the cortex. Most of our sensory pathways follow this kind of trajectory from the sensory periphery through the thalamus to the cortex. So this is really a general problem to think about and worry about. The particular experiments that I'm going to talk about have recording electrodes in the thalamus, and for those aficionados, in the ventral posterior medial nucleus of the thalamus. If you don't care about that, it doesn't matter. But we record extracellular signals, spiking activity, while we are able to deliver light through a fiber optic cable embedded in the tissue. And what this does, it turns out that we are able to deliver a virus to this part of the brain, and it causes these neurons to express in their membrane light-sensitive proteins. And these proteins, I'll talk about on the next slide, enable you to actually control the membrane voltage with light input. So there's various versions of that. And so we have then the ability to close the loop on this. We can record activity, we can turn that around, and use that to guide light input on the very neurons we're recording from. Okay, so there's some details of this, but I'll keep it quick. For optogenetics, this is a whole field unto itself, where a number of laboratories have developed methodologies to make neurons light sensitive. And the real value of this is that they can be targeted anatomically, so we can inject this into certain parts of the brain, but they can also be genetically targeted, so you can select certain types of neurons and not others, and so on. Unlike electrical microstimulation, we can use opsins to both either excite or inhibit, so you can go bidirectionally potentially. And we can use optical stimulation and electrical recording with little concern for stimulation artifact. So the big caveat here is that we don't have a clear path to humans, to therapeutic uses, but maybe that will come, maybe not. Maybe it's just a scientifically investigative tool. What I'm going to talk about is channel rhodopsin, which is an excitatory opsin that we've used. This is to remind me, there's a really nice review article for the control folks in the audience from Carl Dijsselroth's lab that lays out a lot of different interesting problems and the state of the field. This was from a couple years ago. In terms of the basic control framework, we then have the ability to deliver light, and we have the ability to record from a neuron, acknowledging that with the light we're influencing the activity of many neurons, not just one. But we're recording from a single neuron in this particular example I'll show you. And so you might first jump into an open loop problem and say, well, all right, well it's good enough if I characterize the relationship between light input and firing rate output. There's some kind of static nonlinear function. I can just use it like a lookup table, and maybe I can design an input. I can invert this and design an input trajectory that will give me the desired output trajectory. And that works well in some cases, but obviously in the face of disturbance, and I mean this in the broadest sense, maybe unmeasurable inputs, for example, which is often the case in the brain, but also maybe more broadly thinking about parameters of the system that are changing over time. And so the disturbance causes immediately makes us start to think about closed loop control. So in this case, we have a disturbance entering the system, but then the ability to measure activity, spiking activity of neurons. We then develop the control strategy around instantaneous firing rate. That's a design choice. So in that case, we have an observer that translates the spiking activity in real time into a instantaneous firing rate, which we then compare with the reference trajectory, and the controller operates on this error signal. And for the simplicity, we start with simple PI controllers. There's lots of more sexy things we could do. We have all sorts of ideas on this, but this is a good starting point. So just in terms of the signal chain, there's, in real time, extracellular recordings, A to D conversion, spike sorting in real time, which is tricky, and then the observer. And then the observer is compared to the reference trajectory, the controller is implemented, and so on. So we've got to turn that around on the order of 10 to 20 milliseconds if we can. Okay, so that's a real constraint on the design. So just a quick couple of results to talk about. One is, this is a 1 Hz sinusoid. In green is the reference trajectory. That's our goal. In solid black here, the dark black is the closed loop control. And that's the average across many trials, repeated trials, experimentally. The individual trials are shown in light gray. For the open loop shown in the bottom here, we have again the green trajectory is the target, and the thick red is the open loop version. So it's not so bad, right? But one thing to note immediately is that the variability from trial to trial is significantly higher, and that's one thing we've observed in a number of different settings, that the closed loop control seems to really clamp down on variability, and so we get very tight control of the variability. So as you might guess, though, the open loop fails when you start to introduce a disturbance. In this case, it's a whisker input, just to demonstrate what this can do. So whisker stimulus starts at 2, so wiggling the whisker, driving this pathway. The open loop system falls apart. The closed loop control compensates for that by backing off on some of the input, and you get this tracking of the sinusoid. Okay. So this is a more naturalistic input. This is the green here is derived from recordings we took in the awake rat, moving their whiskers out in the world, and then that's the target. The red shows the open loop controller as we inverted this kind of simple static nonlinear function, and the darker line curve here shows the closed loop version of this. So you might say, well, that's not so bad. One interesting observation, and this is the best example where the open loop actually matches relatively well. One interesting observation that's consistent with what I said before is that the variability, again, for the closed loop system is significantly reduced. This is the Fano factor, which is the ratio of the variance to the mean. The Fano factor is significantly reduced for the closed loop control system. Okay. And that's consistent across a lot of experiments that we've done. Okay, so let me dig in a little bit more. And let's talk a little bit about the control setup here. One thing to point you to is that we did publish one paper on this in 2015 with a collaboration with Steve Potter, partly in vitro experiments, but our piece of this was the in vivo. And one of the things we experienced in the neuroscience community was pushback on calling this closed loop control because, or at least the novelty of the closed loop control, because in the neuroscientist mind in general, being in the middle of an experiment and dialing up some light level in the experiment to drive a fiber optic cable in the brain, that was closed loop, right? Meaning that the experimenter's in the loop and that's a closed loop. But we mean automatic. We had to explain repeatedly. So I'm just warning you, when you're trying to publish in this community, you have to be very precise with your language. Don't make any assumptions. So there's two parts of the problem. I won't focus on the observer. It's a kind of it had some hidden landmines in it, but we solved that. It's a little less interesting, and I'll focus mostly on the controller. But just to suffice to say that the observer in this case is really simple. Single parameter exponential filter with a single parameter tau, and we adjusted that to minimize the mean squared error of the reconstruction of the instantaneous firing rate. The more interesting problem has to do with the controller. And so we built, in the earlier publication, it was all very ad hoc. We got in the middle of the experiment, we started turning knobs on the controller gains. We wanted to be more principled in this case. So we first start the experiment by presenting a white noise light input to the plant, measuring the spiking output, and then fit a model to this. And this is a simple linear nonlinear cascade. For those non-neuroscientists, for whatever reason, the neuroscience community feels more comfortable when you show a linear filter reversed in time. So these are impulse responses, but they're just flipped in time, followed by a static nonlinear function that has this kind of logarithmic form, and we fit this with maximum likelihood. So we do this during the experiment really quickly and fit this, and then use the model, which ends up looking something like this, a nonlinear function, linear filter, cascade. And we use that to then design the parameters of the controller. And the goal is, at least initially, was to minimize the mean squared, integrated mean squared error here. So one of the things that we observed here is that as you might guess, for the people into the details of this, that the integral component of the PI controller will bias towards the DC. And so you fight that, right? Because the controller really wanted to track just the DC and didn't care about the modulation. So we ended up weighting the DC and the modulating frequency component and had a variable weightings on those and we tuned that manually to try to balance out the bias between these. There are probably more sophisticated ways to do this. I'd love to hear some ideas. We did this to then optimize this balance between the DC and modulation, and here's how it looks. This is a 5 hertz sinusoid clicking along. The target is in green. Again, the black line is the experiment. You see the control effort that goes into this. In a majority of the cases, the closed loop control did about as well as you might expect based on the LNP, the model fit. So you fit the model and implement that in simulation. It does this well. The closed loop control in the real experiment does like this. The open loop, in some cases, actually performed remarkably well, but in some cases fell to hell. Just completely fell apart. In terms of the naturalistic inputs, this green shows the desired trajectory. The black again is the experiment and the control effort. And again, the model did the closed loop control model did. The closed loop control strategy did as well as you might expect compared to the as well as you could possibly do compared to the LNP model. And then the open loop just didn't do really well at all. Okay, so I wanted to get to this, what we thought was an interesting thing that opened up here. So the model's being used to guide the design of the controller. And obviously there are more sophisticated things you can do here. But one question we had is, well, how good does the model need to be? And one of the things that I would like to share here is that modeling in neuroscience is actually really, the models are terrible. And in fact, one of the things that's really interesting is that there's a lot of model building, but not a lot of model testing. So you read a paper, they develop a very complex model, but in terms of evaluating the actual output of the model compared to experimental data, you often see that step skipped for whatever reason. It's not clear. And when the models are compared, we find that many models, even in the most well-studied parts of the brain, are just terrible. So just brace yourself for that. So one question is, how robust is this kind of control in the face of having a really bad model, or potentially really bad model? So one of the things we did is we said, well, okay, what happens when we look at the closed-loop control system? This is experimental data. Green is the target. The darker line here is what the control algorithm the controller did. And when we replay the inputs to the model, what we found in red is that the model was a terrible predictor of the actual activity. Okay, so the model's terrible. In many cases, just really a bad predictor of the actual activity. However, when we take and this is probably not going to surprise a lot of people who are down to the details of this kind of control design, when we actually plugged in the LNP model as opposed to the real system into a closed loop control, you get pretty good tracking. So the idea is potentially that the model of the plant can be off, and yet your controller can still do a pretty good job of this. But how far can you really push that idea? So we decided to track that parametrically. So we put in a gain and an offset, and we evaluated. You design the controller given the base model system with the gain of 1 and the offset of 0, but then run the simulations while varying these parameters and see how far off can you really be and still track. And so this just shows a summary of that, where in closed loop, this is the offset term and this is the gain term. 1 indicates where you started, so it's the template. So if the model is the plant, then you do really well. But how quickly does it fall off when you perturb the parameters around that? The blue shows the closed loop system is relatively robust to that. You can go pretty far out with the offset and with the gain, but it does fall off. Whereas the open loop system really falls off pretty dramatically if you perturb the system a bit, as you might expect. Okay, so finally, we now have some sense, at least, of the robustness of this kind of control implementation experimentally to model mismatch or model or bad model fits. But there are also scenarios where we can imagine that would be really dramatic changes in dynamics. So in brain state changes, for example. And so what we wanted to explore is the idea that, well, we can't just rely on the closed loop control to compensate for a bad model. In many cases, maybe you need to develop a more complex model. So in this case, this is pretty fresh, just in the last couple of weeks. But the idea is that, and this has some place in the control literature, where we might be switching, the plant may be switching between two different plants, for example, this kind of switch system dynamics. And the challenge for us then is to A, identify that and distinguish that from just having a complicated system, and B, during the course of an experiment, be able to identify which state I'm in and have a control strategy for that and switch around. There's other ways you could do this too, I'm sure, many ways. So I just want to point out that there are lots of examples in neuroscience where things are changing in pretty complicated ways. For example, cortex changing from desynchronized to synchronized states, up-down states within the synchronized state, burst tonic firing modes, et cetera, on lots of different time scales. So I think this is really the ubiquitous in these pathways. So an example of this is, here's one from the visual pathway where local field potential is measured in cortex. This is in cat. Flipping back and forth between so-called up and down states. And a visual input is provided at some point during the course of this. And it turns out that the evoked response, as measured by multi-unit activity in the cortex, is quite different, depending on whether the input comes along during an up versus a down state. And it's different both in gain, so it's larger in the up state, the response is larger in the up state than the down state, but also in terms of latency. So what consequence might this have? Well, there's examples from our lab and other labs where the ongoing activity, for example, in a rat doing a detection task, it has to tell you, yes, I felt my whisker move or not. When you parse the data based on hits and miss trials, this is thalamic recordings, the hit trials tend to have a lower baseline firing rate prior to the delivery of a stimulus. So when you just split these out and say, when did the animal get it right, when did the animal get it wrong, they tended to get it right more when there's a lower baseline firing rate prior to the stimulus delivery. So there are real, not only neuronal consequences to this, but there are perceptual consequences to being able to track this kind of thing during an experiment. So our first kind of nascent take on this is to first of all assume that there's two states to worry about. And by state, I mean brain state, not to confuse it with state in the control sense. But then flipping back and forth between this, we need then in a time course of, let's say, 100 seconds to be able to label different parts of the experiment as coming from one of those states or the other. And so I won't go into the details of this. I'd love to talk about this offline with folks. But the idea in this case is a hidden Markov model structure where there's two states flipping, and you have to infer that from just a sequence of spiking activity. And so the estimation problem, or the hard problem here, is that we need to simultaneously determine when I'm flipping back and forth between these states, and then also what are the parameters, lambda 1 and lambda 2, that dictate the mean firing rates in these states. And so we use an EM algorithm to jump back and forth between this and solve this problem. So that's really nascent. One final example here. Do I still have a couple minutes? Okay. Is an example, and I alluded to this before, sometimes when the dynamics change in some way that the gain changes, it's not actually a hard problem to deal with. The controllers will compensate for the gain, no problem really, in many cases. When the dynamics fundamentally change, for example, this is from the auditory cortex where over one course of the experiment, the impulse response exhibits very different dynamics from just seconds later. That can actually, this kind of latency or these kind of shifts in dynamics can have a really dramatic effect on the ability to control. So what Adam Willits did in my lab is systematically vary the gain and the latency in this kind of set of simulations and switched back and forth between two different systems. So as you might imagine, we're going to assume, let me back up. We're going to assume for the moment that the setup is like this. There's a system here, and there's two systems that are going to switch back and forth, the red and the blue. There's a controller we need to design, and there's an observer. And we're going to assume for the moment that we have a really great observer, so we're not going to deal with that problem for the moment. Let's just say we have this, and we're really talking about a switched linear system, which I think makes the problem a lot simpler. Adam then took those filters, turned them into the state space representations, and used simple LQR design for this kind of control. The real story is, as Steve alluded to several times, there's always like a backstory. Experimentally, we're having trouble implementing the more sophisticated controls than the PI control with the system we have. And so I think this is really where we want to go, and maybe more sophisticated things than this, but right now we're kind of stuck in hardware land. But anyway, LQR design for this kind of control, and this just shows a simple naive thing. I don't know there's two systems. I'm just designing around the average of this. And so green shows a simple static I'm trying to clamp the firing rate of this neuron, but then I'm in the face of this switching state. So blue, red, blue, these are these states switching back and forth. And at every switch, as you might imagine, the controller can compensate to some degree, and the game we're playing here is that the two systems are just changes, they're just gain shifts in each other. So the controller does a pretty good job of this. It's a simple controller. However, when we acknowledge that there are two systems and we're able to switch back and forth the control, so simple just determining what state I'm in and using the controller associated with that, we then can clamp down on these kind of transient errors and significantly improve them. But again, this is kind of preliminary data. More interestingly is when we have latency shifts, or just fundamental changes in dynamics. So red versus blue, I'm switching back and forth. When we switch states here, not acknowledging that, the controller falls apart. Right? It just doesn't track at all. However, when we, and in this case, we've assumed knowledge of which state we're in, so that's only solving half the problem. We then implement these two controller system, we can actually track that pretty well, so the system switches over to the different controller that's been designed appropriately for that plant. Okay, so there's all sorts of things that we can imagine doing from this. So I just want to zoom out a little bit and remind you a couple of the results here. The closed loop outperforms the open loop, luckily for us, right? But that's maybe probably an obvious thing. But I think what's more interesting here is the concept of variability, which may serve an important role in the brain and how it functions. And the way I think about this is the closed loop control has significantly reduced the variability. We can always add variability back in if it's important, right? But you can't take variability away, right? So we've clamped down on this variability and we have better control of that. The closed loop control is robust to model mismatch to some extent, and we've not fully fleshed that out yet, but the small mismatches can be handled. And then control of state switching, I think we're just scratching the surface of this. This is pretty recent, but I think there's a lot of problems to work on here. And all the results I showed for that last part were simulation, but we're digging into the experiments now. And I think this opens the door to a lot of extensions. So I mentioned bidirectional control. So this is with an excitatory opsin, some of the optogenetics work I showed you. There are inhibitory opsins. And we've done a couple of experiments where we get co-expression, so we can put these two viruses in there, get neurons to express both the excitatory and the inhibitory piece. And so then you get full push-pull, right? With electrical microstimulation, you really get, first of all, you activate everything, and you only get excitation to a large degree. So in this case, potentially, we have two directions. They have different dynamics, right? So imagine your push and your pull have different dynamics. That's a really interesting control problem that I'm sure someone's worked on a variant of that. I've talked about a single neuron and a single optic fiber. The technology is there for recording from lots of neurons, but the light delivery and stimulation is really lagging behind this. Our actuators are terrible. They need more attention. So for those of you building this kind of stuff, please work harder. Ultimately, we're moving to behavior, right? The proof is in the pudding. If you can guide behavior and shape perception. And then we have spent most of our time in the last 20 years working the thalamocortical circuit. And so we've done some very pilot experiments where we're putting closed loop control around the thalamus and recording downstream in the cortex. And so I think that's just opening up the possibility to, for example, actuate the thalamus and record in the cortex and drive the control around this loop. And there's lots of experiments that we could imagine doing around that. So finally, I'd just like to finish up by, again, acknowledging the students that did the work. Michael Bolus did the control, the simple PI control at the beginning. Adam Willis did the state stuff at the end. And this is, again, collaboration with Chris Rosell. So thanks very much.
We have time for a couple of questions. Steve, we can wait for the mic. We've got it coming back. Oh.
Sure, great talk, Garrett. So before I ask you a question, I owe a comment for our WashU colleagues. In 1936, George Bishop published a paper from here showing the cyclic excitability of rapid visual cortex. And nobody remembers George Bishop, but if you look at all the Erlanger and Gasser papers, he was their engineer. So these two characters were neuroscientists. They got very famous for nerve conduction. They couldn't have done their work without the engineer techie in the middle. And in the Nobel address, they really ignored this guy as much as they could.
Like Kenneth Cole in the
Yes, he's
in
like
the
Cole.
Hodgkin Huxley.
Exactly.
Yeah.
But my question for you, when you're doing this, the iterative function switching back and forth, can you just run, assuming your CPU runs fast enough, can you just run both models and then do something simple like looking at 1 or 2 step prediction error and just
And use whichever one.
and
Yeah,
just let it
yeah.
run free like that?
Yeah, that's a great idea. So we have not done that. We're a little bit limited in what we can implement in hardware right this minute, but we're switching. But that's a great idea. And we thought about maybe different variants of that, but not quite put that succinctly. But I think, yes, I think that would be a good way to go as well. I mean, that's a very practical solution, right? Yeah.
We have time for one more.
So when you see how this baseline firing affects hits and misses of the animals, do you know what it is? Is it physiological state, level of arousal of the animal, or what do you
Yeah, so we haven't, that's a really good question. We at present, we don't know what it is because we didn't do other measurements that would help us link. There's a weird resurgence of pupil measurement, believe it or not, in animals during experiments.
So that points to arousal, I suppose.
That's right. And so that turns out to be a signature for arousal. So it's potentially related to that. However, these animals are really highly trained and they're jacked up and ready to go. And so they are not, sort of lazing around sleeping. They want the water bad. And so in general, we stop the experiment once we notice that their attention started to wander in a major way. So I don't think it's any sort of, they just stopped working, whatever. There's no trends in the data like that. It just may be that during, they may be thinking about lunch for a moment, or I don't know. It's a good question. There are many unobservable and unmeasurable signals in the brain that we have to contend with.
Would we imagine that there's a small and very well precise number of brain states, or do you think there's a million brain states?
If you just try to answer that question by reading the literature, you will go clinically insane because the literature is so complex over 60 or 100 years. I think there's probably a continuum of these, and I think when we say it switches from this to that, maybe there's a large number of discrete states, and I don't think there's infinite, but not 2 states certainly. So I think there's a lot of complexity to answering that question.
All right. Well, let's thank the speaker.